Good morning, everybody. As you join us for our UK Spine first virtual seminar, I can see the numbers ticking up in the participant box, so welcome. As we've already said in our chat, please do introduce yourself on the Zoom chat. So while you're introducing yourselves, I'm just going to start with the general housekeeping for this morning's seminar. So as you can see on this slide, what we're asking is that as we do our presentations today, you will join and mute your sound, please. If you don't, Brian or Vicky will remind you. To help with bandwidth and to make it the best possible session that we can, we're asking you to turn your videos off during the presentations. Today we'll have 3 presentations, which I will introduce shortly, and we'll have them one after another. As you come up with your fabulous questions for the panel, if I could ask you to put them in the chat section of the Zoom screen, and Brian and Vicky will be collating those during the sessions. Once all of our speakers have finished, we will pick those questions that we see occurring most frequently, and we'll start to pose those to the panel. This is just a note to say that today we're going to be recording this session, and after the session, we will be putting this on our YouTube channel. So if you can't join us for all of the session, or you have a colleague that's been unable to join us, please do let them know. We may well have more questions, or I hope we have more questions than we are able to answer today. If that is the case, we are going to pose these questions to our panel afterwards, and we're going to put a transcript of this together and make this available via the UK Spine website afterwards. So please don't worry if we don't reach your question. We will make sure that we get an answer for you. And I think that is everything that I need to say to you in terms of housekeeping. I've been watching the chat scroll along and the participants go up. So I think what I'm going to do now is start with my introduction. So we'll just move to our second slide, please, Brian. Okay then, thank you. So this morning, first of all, I'd like to welcome you to our first UK Spine virtual event. We had hoped to welcome you all to our second conference on the 2nd of April in Birmingham, and we were unable to do that. So we've decided to continue to bring you some of our great advances by a series of virtual sessions. Many of you are going to be familiar with the UK Spine and our work. We're a Research England funded Connecting Capabilities programme. For those of you who are less familiar, our work is to connect the excellent work that is happening across the United Kingdom to look at how we can innovate and discover new therapeutics and better and affordable solutions to meet how we extend our health span and reduce the morbidities that we live with. And we do this not just by looking at the excellent science, but looking at how we're exchanging and creating new knowledge. We have a range of funding streams and a range of nearly 30 projects running across the UK Spine now. And today, we're absolutely delighted to welcome 3 of the leads on those projects. Our key leads who are speaking today will give a short 10-minute presentation, which they will finish with some of the key opportunities and challenges they see in delivering this new sector. So first of all, I'd like to introduce Ghada Al-Sala. Dr. Ghada Al-Sala joins us as one of our proof of concept project leads. And we're just going to take down these slides now while we put Ghada's slides up. Ghada is a postdoctoral researcher with Katia Simon, and I believe joined Oxford University about 3 years ago. Katia and Garda are particularly interested in studying the influence of immunology and autophagy in aging processes. And this morning, Garda is going to introduce a project looking at targeting one of the transcription factors involved in lysosome and how we can look for new targets in the treatment of osteoarthritis. Without further ado, I'd like to hand over to Garda to present some of the work that she's been doing. Thank you, Garda.
Thank you, Beth, for the introduction. Hello, everyone. First of all, I would like to thank UK Spine to fund my project and to give me this opportunity today to discuss it with you. As Beth mentioned, I'm interested to develop a drug targeting a protein called TFIB for the treatment of osteoarthritis. But let me first start to talk why the area of aging research area is important. As you all know, one of the main transformation of our society today is the increased number of people having more than 65 years old, as you can see here from this figure. But this actually great achievement of the increased lifespan is affected by the increased number of people who are suffering from one or multiple age-related disease. And this is open in front of us a new challenge to understand the aging and the process of aging. And so far, the scientists do have made great effort to explain and to understand this mechanism, proposing to us a model of 9 hallmarks of aging, which we believe and we know that these contribute to the aging phenotype and the aging process, which I will summarize to you here in 3 different categories. The first one, which we believe that might be the cause of the aging, and it's the molecular alternation of a genetic and epigenetic factor. As a response to this alternation, we will have the cellular dysfunction, such as the loss of the proteases or the mitochondria dysfunction. And as a final response to all of this, we will have the tissue degeneration, such as the cellular senescence or the stem cell existed. And actually, all these factors together participate in increase the risk or even the development of several age-related disease. And we believe today that instead of targeting the age-related disease directly, if we were able to target these factors, we might be able to prevent or stop age-related disease. And following this hypothesis, recently in our lab, we identified alternation of one molecule called TFIB. TFIB is a transcription factor which control the biogenesis of autophagy and lysosome. These are the main degradation and recycle pathway in the cells. What we find that TFIB is reduced with age, and this will lead to a cellular dysfunction by the inhibition of autophagy in the cells, and this finally lead to immune senescence. So the immune senescence is what we refer to a decline of theory of function of the immune system during aging. And the immune senescence lead to a higher susceptibility to infection and also poor response to vaccination. And you can understand how this phenotype is important, especially in the pandemic of COVID-19, which we are living today, and that how much these the elderly group are the most affected, and we are hunger to find vaccination which work, especially in this group. So what was really interesting in our work that we were able to reverse this aging phenotype, this immune senescence phenotype, by inducing autophagy using a molecule called spermidine. So spermidine is a natural polyamine. Our cells could produce it, but we also be able to take it from food, as you can see here in this figure. The natto beans, which is pretty disgusting, but you can't believe me, it's very good for your health. So spermidine have been used for a long time to induce autophagy. However, the mechanism of inducing autophagy by spermidine was still missing. And recently in our lab, Han and Zhang, who was able actually to identify this mechanism. So what we know that spermidine is important for the hypocyanation of one translational factor called afield, and the hallucination is important for the translational of protein having a polyproline motif. And Helen find that one of these protein is TFIB. And as I said before, TFIB is so important for inducing autophagy, and this autophagy have its own impact in the immune system response. So what I find actually, which was also interesting, that the whole pathway is reduced with age. So we find that spermidine is reduced with age compared to young, and afield, the hallucination reduced, TFIB reduced, and autophagy reduced. Of course, I'm talking about the BBMCs, kind of white cells taken from the blood. But what was really striking in this all that TFIB was dramatically reduced with age in the blood cells. As you can see here in this figure from Western blot technique, that we could easily distinguish people who have less 65 years old than those who have more than 65 years old. So we thought that TFIB might be a new aging biomarker, especially if we can do this in the blood, which is the easier tissue we can actually extract it from healthy and from patient. But I want to study TFIB in a very specific context of aging, of age-related disease, and so I decided to look at it in osteoarthritis for several reasons. That the osteoarthritis is the most common form of arthritis in the UK, and we know that aging is the main primary risk factor for osteoarthritis. And for the moment, we don't have any therapy for this disease. And osteoarthritis is characterized, as you can see here, by the degradation of the cartilage. And I found if I take the cellular residence in this cartilage, which we called it the chondrocyte, found that these cells have a lower expression of TFIB. And when we put them in culture, the expression of TFIB reduced in the cells, while the expression of one marker of the senescence, which we call the beta-gal, you can see it here, is increased. Adding to this, that there are some recent studies showed that if we overexpress TFIB in the joint of an animal model of osteoarthritis, we could clearly improve the degradation of the cartilage, as you can see here, between the control and infected with the overexpressed of TFIB, and also increase and improve, that synoviocyte scores. So taking all this together, we thought that TFIB might be a very good drug candidate for treating osteoarthritis. And to develop a drug, we have 3 different aims. The first one, we wanted to identify target which could regulate the expression of TFIB. And to do so, we wanted to do a genome-wide pooled CRISPR screen, which are tools. We use it to modify the DNA. And I wanted to do that specifically in a primary cells and not in cell line, in a fibroblast isolated from the patient. And we wanted also to develop a new assay to detect TFIB, because it was so important for us to success this method and to find the right candidate to separate easily the positive cells for TFIB from the negative cells. So we also validate this assay. And as you can see here, which I can't see it now because I have the other photo, this is the number of the flask of the cells that we prepared just before the lockdown. And I hope when the laboratory will be open, we would be back and have some, could actually analyze the data. The second step, and it would be to do identify drug that increase TFIB expression, and this depends specifically in the gene that we will have obtained from the CRISPR screen. And after that, we will validate the effect of these targets in vitro and in vivo. So as you can see here, the impact of my work would be to identify drug based on TFIB pathway to treat osteoarthritis. And this drug might have also great benefit, not only in osteoarthritis, but in other age related disease. And I will finish with one question, which I always wondering, and I hope that Alex and Graham might be able to answer this. If I was successful to find a drug, then how I would be able to move from early translational research to a commercialized and a clinical trial. So at the end, I would like to thank Professor Cathy Simon for her enormous support and all the amazing group of our, the member group of our lab. And I would like to thank you all for your listening. Thank you. I hope I was in the time.
Thank you so much, Ghada. That's fantastic. Really exciting to hear about one of our proof of concept projects. I can't wait to see those flasks full and the lab back in full functioning and capacity. So thank you. If you have questions for Ghada and you're worried you're going to forget them, do pop them into the chat function now. So Ghada finished there by throwing the gauntlet down to our next speaker. So our next speaker is Dr. Graham Wilkinson, who's Head of Virtual R&D at the Medicines Discovery Catapult. Graham, as I should have said at the beginning, you know, we're talking today about how we take things from bench to bedside. Next in that translational pathway of how we develop new therapeutics is translating those fantastic early concepts and proof of concept discoveries that people such as Ghada are working on and make them into something that can be developed on further and assessed as to its feasibility as a new therapeutic. And this is really the work of the Medicines Discovery Catapult. Graham and the team at the Medicines Discovery Catapult have a portfolio of projects with the UK Spine, and today I'm delighted that Graham's going to talk about the first of those, which is looking at a framework of how we develop anti-aging therapeutics and geroprotective therapies in a time where it couldn't be any less important. So Graham, I'm going to hand the floor over to you.
Wonderful. Thank you very much for that very kind introduction, Bev. And Ghada, thank you for an absolutely fascinating presentation on your research. As Bev has said, we at the Catapult are working on a number of projects, and one of these is to try and develop a framework to help the basic research that of the likes that Ghada's prosecuting, to help that kind of move forward in a way which it aligns with the needs of drug discovery. So one of the projects that we're doing in the Catapult is to bring together that kind of framework and to tie together some of the issues and points that are critical to achieve a greater understanding of the requirements of drug discovery. Before moving on to that, I just wanted to spend a couple of seconds just giving you a brief introduction to the Medicines Discovery Catapult. We are one of the catapults that have been set up by Innovate UK in response to areas of high scientific and technological need. We're based in the northwest of England at Alderley Park, and we have fantastic facilities occupying some laboratory space there with a dedicated team of scientists and informaticians. And our role is to support UK life science and drug discovery activities, providing gateways to expertise and resources and access to hard to access scientific capabilities. And we're one of the 5 member partners of the UK Spine organisation. So I guess to build on what Ghada started to talk about, a bit of background about ageing and age-related diseases. So as we're all aware, it's happening to all of us. Aging and age-related conditions, you may be aware, are probably the leading cause of death, morbidity and mortality worldwide, and are associated with some of the greatest costs for healthcare worldwide. But we don't often, as has been pointed out to me by colleagues, go to see our physician or our doctors because we're aging. We go there because we have conditions which are related to the aging process or morbidities as a consequence of aging. And we often develop further morbidities or so-called multimorbidity. And these age-related conditions are the largest cause of or consequence of morbidity, death, and healthcare costs. And increasingly, as we see rises in diseases such as type 2 diabetes and obesity, we expect to see these types of morbidities and multimorbidity becoming increasingly prevalent in younger populations. So there's a real need to understand these processes and find ways of intervening with them in order to prevent, stop, or halt that increase that's expected in morbidity and multimorbidity. There is intense research in trying to understand the aging process. We've heard a little bit about one of those projects already, and this is a worldwide endeavor. And there's many studies have been developed both preclinically and clinically in order to understand the basic processes and mechanisms and hone down on targets which might be of therapeutic utility and those which might be prosecuted in a drug discovery sense. And so the development of new drugs to slow or alter the aging process may ultimately have a dramatic effect on those healthcare needs and the associated costs. However, even though there's been a really substantial increase in our understanding of these processes, much of that has yet to translate into the development of a robust pipeline of new drug discovery projects. And we've seen relatively few investments in new R&D programs in the industry. And primarily much research has been focused on what's called drug repositioning rather than new drug discovery activities. So what happens when we age? You know, how are we going to understand more about the aging process and where we might intervene? So it happens to all of us. We're all getting old. Many of us may engage in more or less unhealthy lifestyles. And as a consequence, what we often see is a reduction in quality of life and our health span over time. We see an increase in the signs of aging at an earlier age, potentially, and that can then be related to the rise of those morbidities and multimorbidity. Currently, what this means as well is that we are taking more and more medications and give rise to the concept of polypharmacy, whereby we're taking many medications to tackle some of these indications. What we want to do, though, is to develop new therapeutics. And as we're all aware, going from left to right along this scale, so targets through to a launch product, is both a time-consuming and expensive endeavor. And most projects fail. We hope to prevent more of that failure preclinically, but still projects go into the clinic and fail there. But what's really important in the context of an aging population is to consider the question of relevance to the patients we're talking about. And that's one of the most important issues we need to think about with respect to developing new and safe therapeutics in an aging population. So we need to consider who we are treating. Often this will be an aged and already ill population. And when are we treating them? I.e., at what age and on the background of what other indications? From that, we need to develop testable hypotheses and the scientific evidence that will be able to link that between the early stage research and the clinical setting. So some of the real challenges that we feel we need to address are in identifying novel targets and mechanisms, understanding the safety and efficacy of those, the regulatory backdrop, because clearly none of this is done in isolation, and then clearly the commercial and economic imperatives which will drive new investment into these areas. So each of these is absolutely vital to the development of an economic case to help investors come in and fund new research, having identified targets and mechanisms which are safe and potentially efficacious, but also which conform to regulatory standards that are currently adopted by the pharmaceutical industry. Once we've got that, we need to consider a line of sight to generate a so-called platform of evidence through to the clinic. And drug discovery, as we as we noted, is a long process and requires many different activities, and requires the generation of evidence all the way through in order to keep ourselves on track and to make sure that we are generating information which is relevant to the setting that we're generating that in. So going from the target right the way through to commercialization, we have to be mindful of the need to progress through this in a way which is both stage gated and meets expectations both of the regulators and healthcare providers. But what are we going to do in this? How are we going to identify new targets and intervene in those aging processes? So, taking these figures from work that in the literature, there are a number of hallmarks of aging which have been alluded to already, and a number of consequences of those going wrong and places we can intervene in. But as I've mentioned, there's this lack of research funding in this area. And we undertook an analysis of clinicaltrials.gov earlier this year, looking at studies in aging. And of those working through a triage, we came across the startling fact that although there was much research going on clinically and interventional studies, nearly all of those were looking at existing molecules, and none of them were for investigational new drugs, underlying the principle that we're mentioning here, that there is very little going on at the moment in this discovery setting. So how do we focus our efforts? Where should we take ourselves forward? As I mentioned at the start, we have aging as a natural process. We're beginning to understand some of the processes and how they relate to the development of morbidity and then subsequent morbidities. And it's our contention that we should be focusing on how we can develop and show that clear mechanistic and translatable link, which allows an investigator to show a testable and develop a testable hypothesis, and with that develop clear strategies to stratify patients and come up with clear biomarkers which allow us to progress forward, relating those processes of aging through to these morbidities and the development of subsequent morbidities. Not an easy task, and we're fully aware of that, but through the work that we and others are doing, we hope to be able to align these ideas and develop these biomarkers and new drug discovery projects. So where should we focus, we believe, to unlock this potential? So clearly aligning those targets to disease, biomarkers which allow us to test the hypothesis. There's got to be a lot of work done, we believe, in understanding the best regulatory framework for this, and that will allow us to have really, I guess, to address these questions in clinical trials which are relevant to the patients that we're trying to test. And on the back of that, there's got to be a clear health economic and real-world evidence-based case to generate the commercial viability of these opportunities, to unlock the funding from pharma sponsors in order to get that investment going. So to move back to Garda's question on how do we move from early stage translational work to commercialization and clinical trials, hopefully I've identified some areas here, but moving forward into Alexander's next presentation, I think one of the clear areas that we need to think about is how do we do more to fund exploratory clinical trials in aging research and multimorbidities. So I'd like to finish there and hand the floor back to Bev and onward to my colleague, Alexander.
Thank you very much, Graham. Really interesting presentation, and I think it's always good to think about the onward translation of science and moving forward from actually proving that we've got something that is tractable, that can be produced, that is safe and effective, that reduces the polypharmacy that we see so often as we grow older. We need to think about the clinical trials, and Graham again has very kindly thrown the gauntlet down to our next presenter. So our next presenter is Alexander Masters, who has just joined his camera on to join us. Alexander is a journalist, a biographer, and the co-founder of an initiative called Eye Cancer. that initiative, rather interestingly, moving away from the pure science of today's presentations, crowdfunded a clinical trial for cancer treatment. And Alexander and his colleague Dominic have come together to work with Heather Draper from the University of Warwick to look at how we can actually assess the feasibility of rolling out clinical trials which are crowdfunded, and how we could do it in the format of ageing as well. So without further ado, I'm going to hand the floor over to Alexander for something a little bit different at the other end of the drug discovery pipeline. How do we be more innovative at getting the right trials to the right people? Over to you, Alexander.
Hello, thank you very much. And thank you also, Garda and Graham. Excellent presentations. I hadn't realized what a rich, exciting field this was. What we need to do is, we're coming now to the third step in that process that Garda set up. We've had basic research from Garda and ways to push discoveries forward quickly and efficiently from Graham. But how do we pay for it? Hang on, I just realized I forgot to share my screen slide. Hang on, here we go. There we go. How do we pay for it? Is the next question. And specifically what I want to talk about with this research project is how do we support promising, high quality, peer-reviewed research proposals which have found it difficult to secure funding from the usual sources. So Bev has already explained I'm not a dentist or a medic in the pharmaceutical industry. My job is a biographer and journalist, and I got into this field because a friend of mine had pancreatic neuroendocrine cancer. And I joined another journalist to set up a campaign group. The journalist was Dominic Nutt, to fund a clinical trial of a neglected potential treatment at University of Uppsala. That I'd read about this, a genetically engineered flu virus, as it happens. We raised the money in 8 months, partly through crowdfunding and partly through a nascent version of the novel funding mechanism at the heart of this research project. That's not coming up. Ah, there we go. So what I'm going to describe then is a remarkable collaboration that UK Spine has funded as part of their role to connect capabilities between Professor Heather Draper, Chair of Bioethics at Warwick University, Professor Simon Bowman, a consultant rheumatologist in Birmingham, an experienced member of the Research Ethics Committees, and Dominic Nutt, who, like myself, comes to this subject as an outsider, a member of the public. Heather is the person leading our work, so any foolishness or idiocies that I come up with in this presentation, they're due to me. Here's a slide where I feel Heather's hand guiding me through this work, this talk today. So I'm rather fond of this slide. At the start of my Uppsala campaign, I came up with an idea about a new way to fund clinical trials, which it turns out no one had used before, at least not officially. I've called it committed philanthropy. First, then let me specify what the general problem is. There is a drastic shortage of available funding for early phase clinical research. And so every year, numerous, nobody knows how many, perhaps hundreds, perhaps thousands, of interventions that might have gone on to save lives and ease suffering are put aside and forgotten. It's been estimated that up to 2/3 of promising early phase clinical studies are dropped for reasons that have nothing to do with safety or efficacy. There are many reasons for this appalling attrition, but most of them, one way or another, are to do with money. In the case of aging research, there's another complication. Elderly patients with multiple illnesses and complaints are difficult to study. What's your target? What are your clear endpoints for the research? The usual ideas about how to frame a good clinical trial start to break down. Committed philanthropy is a new way to finance good research that is struggling to get over these troubled, early proof of concept stages of development. I believe it is potentially an extremely valuable source of new money. The idea is simple to state. Allow rich patient donors to pay for entire trials, the whole thing, the research, the regulatory costs, all that tedious paperwork, all the other participants, in return for the right to be a volunteer on the trial. There is nothing like this available at the moment. I think of it as a kind of Robin Hood funding. The donor must both pay to make the trial happen and be shackled to benevolence when it does, i.e. they must also pay for all the other eligible participants. You can read more about the details of the idea here in an article I wrote with with Dominic, who helped me develop the idea. It's in the Journal of Medical Ethics. When I first proposed it, I called it, as you can see, I think on the title there, the plutocratic proposal. It was a sort of an unclear rather, nod to Swift and his modest proposal. I've now renamed it Committed Philanthropy. As any of you who have applied for academic funding will know, the sort of money I'm talking about here is in the low millions, £2 to £3 million, maybe 5 million, let's say. This is not a large quantity of money for very wealthy people. For our Uppsala work, for example, one such wealthy patient wanted to fund 10 similar research projects at about these costs, using a very early version of our idea of committed philanthropy. Immediately with this idea, there are caveats. All communication between donors and researchers under this scheme must be mediated by an appropriate matching agency. The matching agency is the body that ensures the process is ethical, prevents issues such as bullying and exploitation. This matching agency acts in exactly the same way as any other traditional funding agency, for example, the Medical Research Council. Let's say, God forbid, that GARDA's work finds it difficult to get funding to go into clinical trial. As far as GARDA is concerned, she would apply for committed philanthropy money just as she would for MRC money, and her work and her proposal would be subjected to a degree of scrutiny and oversight at the end. The only difference from GARDA's point of view is that for receiving funding for her neglected research, she sets aside one place on the future trial to give to the matching agency, the MRC equivalent, who will pass it on to the donor. Under committed philanthropy, the donor and GARDA are not allowed to be in direct contact at any point before the trial starts. This immediately leads to the second caveat. This patient donor must be a suitable volunteer. She or he must meet the inclusion criteria and none of the exclusion. If, when the money was presented, the donor had been a suitable volunteer, but the donor's symptoms change, so that once the trial begins, they are no longer suitable, that's tough. They cannot join the trial they have just funded. They also cannot join, of course, if they've died. It would take several years for the researchers to do the final elements of it could take several years for the researchers to do the final elements of preclinical work and wade through the paperwork before this trial can be started. But the donor's money will still be secure from the researcher's point of view, whether or not the donor has survived. It can still be used to finance the trial. I argue that committed philanthropy is the only model of research funded by participants that is ethically plausible. In fact, as I say in our article in the Journal of Medical Ethics, in my opinion, it's more ethical than current funding mechanisms in several respects. But I won't get distracted by that here. Yet when Dominic and I approached researchers suggesting that this was a possible new way to fund neglected work, we were told that in spite of it all, research ethics committees might still throw it out. On one occasion, I went to Canada to speak to a group there about the idea. The head of the hospital ethics committee asked me to be escorted off the premises. It was thrilling. The mild suggestion of a way to fund neglected research by getting extremely wealthy people to pay for everyone's participation was dangerous enough to secure me security guards. Clearly, we need to have a better sense of how research ethics committees in general will respond to committed philanthropy. Hence this collaborative project with Professors Draper and Bowman at Warwick and Birmingham. Our collaboration is to study the last point on this slide. And I'll go back to that. Our concern that we have experienced, that this thing that we've come across, that research ethics committees might just not approve this idea, irrespective of any arguments that we put in the Journal of Medical Ethics, or which other ethics researchers have agreed that this is an ethical model, and there have been some papers about that. But nevertheless, research ethics committees would not approve the idea for one reason or another. And that's why we have ended up working with Heather to devise an experiment that would be able to test to what extent this is really true. The problem we're facing, how will research ethics committees, in particular those of the NHS, respond to this idea, can be divided into three parts. First, there's the empirical question. Let's say you have a good proposal for a clinical trial, but you also propose to get it funded using committed philanthropy. What will NHS research ethics committees have to say about that? They're the guardians of the UK's clinical research world. If they reject the idea of this new funding mechanism, committee falls at the first hurdle. For this research question, there doesn't need to be an objection that falls. I've got someone's, I'm overhearing someone. Are you off? Are you on? You're off again. Okay. For this research question, I'm still overhearing. Never mind, I'll go on. For this research question, it doesn't need to be an objection that falls within the ethical remit of research ethics committees. It might be a gut reaction. Committee members are humans, after all, not algorithms. Perhaps there will be a feeling that despite the fact that the purpose of this proposal is to rescue work that would otherwise not be available to any patient, there is something really unfair about the idea, a feeling that it is better to level down and ensure no one gets to share the benefit, rather than one very rich person being in some way marked out. The second question is the more straightforward one, the ethical one. Under what conditions, if any, ought REC's research ethics committees be willing or unwilling to provide a favourable review? As a consequence of these two research questions, a final research question for empirical ethics. In other words, how do we resolve the tensions set up by those first two problems? Heather is educating me as rapidly as possible in the philosophical subtleties at work here. It comes back to this question of how a person on a research ethics committee ought to respond to committed philanthropy, if they were a well-oiled algorithm, so to speak, versus how they will behave as ordinary human beings like you and me. Then imperial ethics asks, how, if at all, should any perceived concerns about this new model be addressed in policies? Our project will proceed. This is it in a nutshell, and we are just at the start of it. We have identified 3 groups we think will help inform our thinking, the research ethics committees, but also we're interested in the views of researchers, because although the RAC's primary obligation is to protect research participants, so they play an important part in pushing medical research forward and protecting researchers from undue criticism. They have a clear stake in the integrity of research ethics committees. Finally, we are interested to know what ordinary people think about the idea, the me's and you's. Any of us might at some future point be approached to join or even be desperate to join a clinical trial. How would we feel about this trial being donor funded? RECs are supposed to consider the balance of harms and benefits of participation from the perspective of ordinary people, and to some extent at least, policies for RECs should reflect the views of the population they serve. Then all being well, we draft policy and guidance documents to present to the Health Research Authority's National Research and Ethics Advisory Panel at the bottom there and publish a nice paper. Where are we in this? Right at the start. So despite having just talked for 10 minutes, I still don't have a lot to tell you about any results. We're at the beginning. We have run a pilot project. Now begins the hard work. But before I go, I want to say something which I feel very profoundly and which I feel, and I think Dom feels the same, moves us both, and that we are genuinely honored to be part of this research. Such true, close collaborations between the academic world and, in our case, the activist world are rare. Heather is the brains behind this ethical research project, the prime mover in terms of making the components happen. But she has involved us at every stage of the way, and we've had an active role in shaping how the research is to be done. I can't stress how much her generosity in this and UK Spine support for it means to us on the outside. Thank you. I also want to thank the VictoryNet Foundation, who funded initial work on committed philanthropy, and Magnus Essend, Dai Lee, and Justina Liga Jarlblad, who are the team at Uppsala, and our co-worker Liz Scarf, but above all, the thousands of patients and activists who made our initial eye cancer funding campaign a success, which is what led to the idea of committed philanthropy. Without them, none of this would have been possible. Thank you.
Thank you so much, Alexander. A really interesting field area and something that I think we're already seeing quite a lot of questions coming through on. So what I'd like to do while we get ready for some questions is to ask the speakers, so Garda, Alexander and Graham, to pop their cameras and their microphones back on for me. And I am just going to scroll through some of the excellent comments that we've been having in so that we can ask the panel some questions. We'll stick to the order that you very kindly presented in, with answers coming from Garda and then Graham and then Alexander, just for simplicity. So the first question that we've got in is coming from Ofer, who I can see a wonderful picture of Ofer. Ofer, if you want to switch your camera on now, that's super too. Ofer's question is, hello, I would like to ask a general question to the speakers, please. I agree, oh no, that's the wrong one, hold on. Let me, sorry. Great talk. Based on your introduction, Garda, what are the prospects for increasing TGF-beta using nutritional supplements that increase spermidine levels?
Okay, so thank you so much for this question. It's really a very important one. So, I mean, we need more investigated to know what will happen. But for the moment, there is a lot of clinical trial showing that if we give a spermidine directly, now you can even buy it, actually, it's commercialized. And studying this in a cohort of elder people. For the moment, it seems to be safe, and we don't have that, problem or side effect of it. However, I just want you to put in mind that this should be like spermidine and TFIB is already in enough quantity when we are young. So if you give a spermidine to a younger person, it will not have any effect. This is what we believe. But of course, we have to investigate this more. I understand also your question. It might be related that TFIB is somehow implicated in other disease like cancer or whatever. So all this should be studied carefully and investigated more. But I believe that the main thing is that we lose the balance. If we don't have enough of the protein and then we could give it over, this will give a benefit. But if you overexpress it and you have enough, then might you have the problem. And I think this is the main problem of aging, that we lose the balance between the damage and the repair. I hope that I answer your question.
Thank you.
Ofer, anything to add from that before I go on to my next question? Okay, super. Well, I can see questions coming in thick and fast. So next up, I'm going to go to a question from Lynn Cox, who is somewhere in the wonderful panel of faces that I've got looking back at me. And the question that I've got from Lynn Cox is, since aging is not classified as a disease indication, why would pharma become interested in drugs that deal with aging rather than specific diseases? Ah, there you are, Lynn. Hello.
Hello.
I'm going to ask that question to Graham first.
Yeah, hi Lynn. Thank you very much for the question. It's a really pertinent one. First of all, I do think there are efforts underway to try and get aging classified as an indication on the next ICD codes. I'm not sure how that's progressing. But in terms of where we are now, I think one of the things we'd like to see achieved from a lot of this research is, by intervening in one process, how that can stall, prevent, or even halt the progression to other indications, which might not necessarily be something you'd go after through a single route. So that could be one of the kind of the motivations in that way. But I think what we might be seeing is that under the current regulations, at least, pharma would perhaps start to get a better understanding of the relationship of targets to a variety of different processes and indications, and through going after one or a small group of those, perhaps in a small combination, be able to bring a product to the marketplace which can be more impactful than just in a single indication. So I think that might be some of the underlying motivations ultimately. But I think, as we've probably all alluded to, there's much work to be done to really uncover those relationships and to identify targets which can impact on more than one specific indication.
Thank you.
Thank you very much, Graham. I wonder if any of the rest of our panel would like to comment on that question.
Yeah, I just want to say that
You go ahead. You're
yeah, thank
you
you.
are the next to speak.
Yeah, I think it's a very important point. But if we, as a scientist, we were working more on showing how this process and mechanism of aging is associated with the disease and the cause of the disease, because for the moment we don't know if this process of aging will cause directly. It's associated with this disease. So more scientific and more work need to prove that if we really control the process of aging, will target the disease, this will make the company and the pharmaceutical more interesting to treat aging, in my opinion.
I mean,
You've got Alexander.
well, I was thinking, just from an outsider's point of view, they'll become interested because I'm interested, and they'll change. If I want this stuff, whether it's classified as a disease indication or not that they're treating, I'm sure the market will change itself so that it can supply me with it. I think that the way we were brought in, Dominic and I were brought into this project, was through this difficulty. How do you deal with the fact that this isn't a disease indication, given that the whole process is set up to treat disease indications and this is not? So funding then becomes difficult for such projects. This is as it was presented to me. Hence why you might need a new approach to funding to deal with this worry about aging not really being a disease indication. But I honestly think if people like me want to get hold of this stuff, because whether our age is disease indication or not, the market will change to do it, surely.
If I could just come back to you slightly on that, it's more getting through the FDA so that pharma can actually sell their drugs. Right. And without an indication, they won't get a license, they won't go through the regulatory hoops.
Yeah, I mean, okay, my response is simply then not, we're coming in terms of committed philanthropy as a possible alternative route forward if such obstacles present themselves. And my comment then is superficial in the sense that, yes, of course, things will change eventually.
Thank you, Alexander. So, I mean, this is, you know, such a hot topic at the moment. We've seen clinical trials by companies such as Restore Bio fail for arguably what are reasons that it shouldn't have failed in phase 2. And if you're interested in that, there's plenty of literature online about what happened there. In the interest of being fair to the whole panel who've given up an immense amount of time to support today, I'd like to come to a question to Alexander next. So I have a question from Anna Ashton here, and it says, "Hi Alexander, fantastic idea. Why limit the donors to people who are patients that can participate in the trial rather than allowing other non-patients to donate as well?"
It's a very good point, and the real answer is for ease of presentation in this talk. I didn't want to get caught up in all sorts of parentheses and additional points. It is absolutely open to anyone who wants to provide funds. What they get back is the option to take on a place on the trial, to volunteer for a place on the trial, and they can award that to whoever they like. They can award it to their child, to their parents, to their friends, to a stranger. They might pick up the case of someone who they don't know at all and decide to allocate the place to that person, should that person still be a suitable candidate. And there have been instances of wealthy people setting up trials in the hopes that it will benefit some someone who's a stranger to them just because they've been moved by their plight. So it's completely not limited. I limited to that in the talk because I was aware I didn't want too many commas and brackets to fill up what I was saying. But thank you, a very good point.
Thank you, Alexander. And I just wonder if we focus on the sort of the modes of committed philanthropy. Graham, I wonder, you know, one of your questions that you posed at the end of your talk about how we exploit different ways to fund clinical trials, or how can we do this differently? Have you got anything to come back to, Alexander, on as to how we can put pressure on industry to be more engaged in this sector?
Gosh, that's a very difficult question to answer, I think. I think industry will take notice if they think they can make a commercial return, and that the regulatory frameworks are open enough for them to be able to move forward in a way which is is relatively straightforward and understood. So to the point that I think was raised by Lynn, you know, having a regulatory framework, ICD codes that you can develop a drug against. So for pharma, I think it's and biotech for that matter, it's got to be about, well, I'm going to do this because I need to make a commercial return. Show me the way I can do this. Help me identify the targets, the really good causal link to disease, and a route through to commercialization. And I think we'll start to see investment there.
Absolutely, and really, that's a good point to say that this is the premise that the UK Spine is working on, in a way that cell and gene therapy disrupted and created a new sector, bringing cure where before there was not. The idea of looking at aging science and the idea of where we intervene in underlying pathways to increase our health span is something that becomes increasingly more important. And I think where I'd like to wrap up today's fantastic conversation is that we're all well aware that we're existing in this new normal, this post-COVID-19 situation. And if, as we've seen and we anticipate that we've just seen a 1 in 50 or a 1 in 100 year pandemic, we could, many of us on this call, be the people facing the next pandemic as that population over 65. And we will be a global population of people who are older rather than younger. And therefore, this is a question of global importance. How do we maintain our health so that we reduce our morbidities, we reduce our polypharmacy, and we prescribe new therapeutics more appropriately to the way in which we need them? And so I think I'd like to finish by thanking Garda and Graham and Alexander for addressing the challenges and the opportunities that we see from basic science in drug discovery through to commercialization, through to creative ways to make sure the right drugs and the right treatments get the right opportunities to reach the clinical trials and the people that need them. I really hope that you've enjoyed our virtual seminar today. We wish we could see you in person, but as we realise it might be a little bit of time before we're able to do that, we'll be having a series of virtual online events over the summer and autumn until we can reconvene with our next conference. So I'd like to close today's presentation by saying a big thank you to our speakers, and a big thank you to everybody who's joined us and managed to stay on the line for the whole of our session. Thank you very much. If you want to unmute and give a round of applause as we sign off, please do so. Thank you.
Thank you very much.
Goodbye, everybody, and we hope to see you soon. Have we